My goal in this paper is to inform several misconceptions regarding the subject of DNA science, which further misinform the opinions of people who absorb this information without fact-checking. In the end, this information is of a general nature and not specific to the subject of Sasquatch, yet this general information highlights some of the problems with the available Sasquatch DNA evidence. I understand that many people are sincerely attached to their personal beliefs regarding the place where the subjects of Sasquatch and DNA intersect. Passion is irrelevant to science, and we all know one thing with certainty. Between where we are now and where we will be in the future, when we understand this subject well, we will all learn new things, and we will all discard ideas along the way. The path to knowledge is littered with the shattered remains of the cherished truths found to be wrong, and the bodies of those who are unwilling to leave them behind. As of the original drafting of this document, in November 2020, there is no convincing DNA evidence for Sasquatch publicly available that I have been able to find. You may hold a different opinion, but that opinion is misinformed. You are entitled to believe whatever you want to, but facts do not support your belief on this topic. There are many reasons people do not fact-check. Many lack the knowledge or expertise to understand the details of this information. Many expect the people from whom they are receiving their information have vetted that information themselves. This is often not true, and often for the same reasons of a lack of relevant expertise. This leads into the idea of convincing people, but this is a false goal. Verifiable facts will do that. There are certainly those who do not believe the Sasquatch exists as a species in the natural world. Set aside the concept of needing to convince those people the species exists. Instead, set your goals on learning what you can about this species in a thoroughly analytical framework of rational and critical thinking, organized around the accumulation of observations that can be distilled into a set of properties that can be used to define the phenomena others can look for and attempt to observe. DNA is just one aspect of this species, one set of phenomena that must be understood more clearly. We understand virtually nothing about these beings clearly, and that is why they are unconvincing from the standpoint of rational thinking and critical analysis. I will make a brief review of known Sasquatch DNA studies to establish some background. This will include other sources of evidence as well, and a brief discussion of how evidence might be assembled. Back up. This will include other sources of evidence as well, and a brief discussion of how evidence might be assembled in a manner acceptable to science in the end, but would benefit the community immediately. I am going to make a distinction here regarding the intent in propagating false information. When I use the term disinformation, I specifically mean deliberate dissemination of incomplete or misleading information with the intent to mislead people into a false narrative. When I use the term misinformation, I specifically mean the dissemination of information not known by the provider to be incomplete or incorrect. Typically, these are well-meaning people who are themselves misinformed on the subject, and I emphasize their earnest belief in what they say, even if it is wrong. They do not have the intent to mislead their audience. For example, when one speaks on a subject and presents false information, their audience leaves misinformed. When their audience spreads the word, they are, by definition, spreading misinformation. They lack the intent to deceive in doing so, unless they have determined the information is misleading by their own fact-checking. If the original speaker intended deceit, I will call their presentation disinformation, but that is challenging to prove, so unless I specifically use the word disinformation, I will presume a lack of malicious intent and presume that they are simply misinformed themselves. I would point out that there is never a specific logical path that requires original dissemination of misleading information to be misleading. An originator of information may not be familiar enough with the subject to realize they are themselves misinformed. This is broadly known as the Dunning-Kruger effect, and this psychological phenomenon affects everyone. This makes it vital for everyone to independently fact-check anything. It is a moral responsibility to ourselves and our community, and to stop spreading information we know to be misleading, and to inform those who do not understand the subject. I do confess that I often fail at this last part. I mostly don't care what people want to believe. If they are open to facts, I will talk to them. If not, I will either bite my lip or I will leave. I will frequently refer to Sasquatch in this document as the subject. There are multiple reasons for this, not the least of which is to use a less triggering term for the skeptical. We do not know what these beings are. We have good reason to expect that something does exist out there. Once well understood, will meet our expectations for a species we often refer to as Sasquatch. The accumulation of phenomena strongly suggests something is out there. We do not know how sophisticated they are, how intellectually advanced they are, how socially advanced they are. Referring to the subjects also helps hold the idea that more than one thing may be going on out there. There have been numerous attempts to sequence DNA from the subjects. Some may have been successful, but this is absolutely unclear. The Sasquatch Genome Project, SGP, was the most visible and controversial of those. A quite thorough analysis of this project is in the book The Sasquatch Genome Project, A Failed DNA Study. I will summarize that while a portion of the published data is interesting, the conclusions published are simply not supported by the data published. I will discuss this more below. Around the same time as the SGP was the Sykes Study. This study identified known species from all the samples that yielded DNA in the study. I will discuss this more below. There have been many others, and in general they all yielded one of two interesting results. Failed to yield DNA from the sample, or sample identified as human DNA. Other results indicating a known species are frankly just not interesting to the purposes of our studies. In the first video of this series, I covered superficial problems with the SGP and my personal path of discovery leading me to dig into the SGP for more detail. To summarize, I had no skin in the game approaching the situation from 2019. While I could perceive the intellectual battle lines, I had missed the passion of the time. I was more of an archaeologist of the situation. For the record, I communicated with Dr. Ketchum after speaking with Mitchell Townsend in early 2020. He said she was earnest and open-minded, which did not entirely fit my expectation from observing the controversy falling out of the SGP study. She is understandably gun-shy of outside contact given her perceptions of how the paper was received, both professionally and publicly. I was honestly not particularly impressed, and I discontinued contact when she entered a loop of conspiracy theories regarding the Men in Black and government suppression of information. From the conversation, however, a number of highly salient points did evolve. No hair sample produced DNA from the medulla. All DNA recovered from hair samples was from tags of flesh at the root of the hair. Thus, they were exposed to elements in the wild and subject to contamination. The SGP did none of its own lab work. All the DNA chemistry was performed by contract laboratories, even though some parts of the process were customized by the SGP. She only published a fraction of the data. She said she has around 4 terabytes of data. She concludes that species identification from mitochondrial DNA is not adequate to identify Sasquatch from human. She says that the laboratory operators were threatened about doing any more work for her project, and that their families were threatened, not just their businesses. She is continuing to seek ways to finish processing all of the samples and does plan to publish all of the information at some future date. The SGP generously published a small fraction of the DNA data they acquired. I believe it to be the most convincing data they produced. Fortunately, the published information points to several issues, all analyzed in detail by Dr. Hart in his book, that demonstrate failures in this process. The fact that she claims to have terabytes of data does not matter. What was not shared does not exist for evaluation. The same is true of information other people hold in secrecy. It may exist, but it is irrelevant to the public discussion. Secret information is not information at all. The DNA from hair samples will be a recurring theme as this paper progresses. As Dr. Hart discovered, a few of the published samples contain such unusual information that more work is demanded by the exercise of good science, if only to understand how these unusual mutations appear across a number of samples in the project. I cannot speak to her conspiratorial anecdotes. I believe her perceptions on the matter, but I do not have convincing evidence supporting the interpretation of her experiences in the matter. My contact with Dr. Ketchum ended after briefly broaching the subject of contamination. She would not discuss the issue until I watched her video on the subject and was convinced of its completeness. I had already done so, and she insisted that it covered all the relevant information anyone needs. I believe Dr. Hart was reasonably thorough on this subject and discusses issues that Dr. Ketchum ignores. It is my opinion that Dr. Ketchum is genuinely convinced of what she professes. The fact that what she presents does not stand up to scrutiny is not relevant to her, but it should be to you, provided your quest is for objective information that can withstand rigorous investigation. What troubles me in this is that I would expect the subjects to be from an earlier point on the phylogenetic tree. Denisovan and Neanderthal mitochondrial DNA are more distinct from ours than any of the samples from Dr. Ketchum. This seems to lead me to a basic choice. All of the SGP samples are human contaminated. Not entirely unreasonable, but why are there these unusual mutations, identical across several samples originating from different locations? Probable contamination by the same human? Or botched contract lab work processing these samples in parallel? Or, just perhaps, Sasquatch DNA is actually more similar to ours than even our closest human ancestors, meaning elements of Dr. Ketchum's narrative is more or less correct, even if accidentally. The Sykes study limited itself to DNA recovered from the medulla of hair samples submitted from around the world. On the surface, the study proved that, while most people who have samples of hair they believe to be from a cryptid hominid are almost always from some known species. Further, that objective examination of the hair in the first place should have contributed to correct identification of the species in question. He did find one sample that yielded human DNA, but I expect that is actually human DNA. His study performed species identification using fragments from the mitochondrial DNA recovered from the medulla of the hair samples. Specifically, the medulla was used because the body of the hair performs functionally as a sealed container, allowing samples to remain uncontaminated for centuries based on forensic studies concluded by the lab that performed the analysis. The lab was and remains the best source in the world for this kind of analysis. The study specifically excluded samples that did not yield DNA from the medulla, and this becomes an important detail in understanding the results regarding the subjects. A number of samples submitted to the study that were genuinely believed to be from Sasquatch were excluded from the study because they failed to yield DNA from the samples. As discussed in the first installment, I first reached out to Dr. Meldrum on the Friday before Christmas 2019. I had reached the end of what I knew to do with the analysis of the SGP data and was seeking his advice for better understanding to discover more paths of analysis. The next morning I received a response with a broken link to the Relict Hominid Inquiry website. It was a paper by Dr. Hart that examined a number of different DNA studies related to Sasquatch. After reading this paper, I reached out to Dr. Hart, and I made a new friend. On a couple occasions, I reached out to Dr. Meldrum again. There is an important point that is consistent in his exploration on this subject. The medulla of hair, convincingly believed to be from Sasquatch, has consistently not produced DNA. Essentially, it comes down to this. If their hair contained viable DNA, this would no longer be an issue in the world of science. We would know they exist, even without a holotype specimen. I concluded that a study of hair from the subjects is a significant shortcoming from the community. I believe there are those who may be able to produce such a work, but they lack the expertise to properly form their data for publication, or they lack the will to share the information they have due to some misguided sense of propriety. There are many options to this end, and people such as Dr. Meldrum and Dr. Hart would be excellent resources to contact. Dr. Hart became interested around the time of the SGP. With all the controversy at the time, he took it upon himself to learn what he needed to understand what was really going on with the SGP data. Dr. Hart is a witness of Sasquatch. For those of us in this category, even for science, it is not a matter of proving they exist. It is a matter of understanding more about them by seeking to observe the species, its behavior, and its place in nature. I have found Dr. Hart to be dedicated to logical thinking, but open-minded to recognize there are things that witnesses perceive of these beings that we do not understand well. He has developed for himself an expertise in the understanding of the science of DNA, and has been very helpful to me in presenting that knowledge over the last few months. What I have been able to glean in my own research on the subject is small. I will wager that there is useful information out there not being shared, yet the people who possess it may lack the expertise or the will to properly present the information they have. Such a shame that the unwillingness to share what might be vital and useful information prevents the progress of real science under the veil of personal intellectual property. I am convinced that there is at least one who possesses a potentially substantial body of information on the subject of Sasquatch hair. That person has not responded to queries and is associated with a project that has a reputation for secrecy and an established record of not having provided any meaningful evidence that the subjects have ever been convincingly observed by the project. What they know does not matter. It is held in secret, and it is of no value to anyone as a result. The hair from mammals has many different forms, and the hair from primates is in general distinct from that of other species in several ways. It can be hard to tell between some species of primates from hair alone, but in general it is possible to determine if a hair is probably from some non-primate species or probably from a primate in this way. Regarding the Sasquatch in particular, the root of the hair seems to be consistently different from human hairs. Human hairs tended to have a bulb at the root. What is believed to be genuinely Sasquatch hair usually has more of a spade point shape. Human hair typically produces mitochondrial DNA from the medulla when one is present, and DNA recovered in this way can be uncontaminated for centuries, even while handled by people, museum curators and visitors, for example. What is believed to be hair from Sasquatch consistently does not yield DNA from the medulla, or, as Dr. Meldrum points out, this would no longer be a mystery. The fact that one sample from the Sykes study did yield identification of human DNA means one of two things we simply cannot prove at this point. This is the first actual sample from the subjects that yielded DNA, though many other samples failed to do so. Or the sample was actually from a human, and this method has been used to identify museum specimens that are centuries old. The best way to know would be to obtain a whole genome sequencing of the mitochondrial DNA from that sample and analyze the data. Only the whole genome sequencing in the SGP data reveals the excessive number of private mutations and the unusual repeated mutations from those samples. Science does not work in secret. Secrecy is for business and for politics, and a quest for personal notoriety. In the end, no one will own Sasquatch. However, people are deserving of recognition for any intellectual property and evidence they bring to the community. Intellectual honesty is required of the community to bring this information together, and a lack of trust here is both the greatest barrier and well deserved. With open science, the attempted theft of property will be apparent and legally enforceable. The problem is not just theft of intellectual property, it's also sabotage of research areas in various ways. Not always malicious intent, but destructive interference with a researcher's specific approach. Real and convincing evidence for peer-reviewed publication simply does not exist at this time. If it did, then there would be published papers about the species. It's not a matter of a conspiracy, it's just a matter that the story is, as yet, not complete enough given the current state of information. We have trackways that are reasonably convincing. This has become Dr. Meldrum's specialty on the subject. Details in some casts tend to cast doubt that the hoaxers would be that sophisticated in biology. Details in trackways themselves indicate behaviors that tend to cast doubt on hoaxers as well. Still, tracks alone are not a complete enough story. While there are not peer-reviewed publications on tracks, the preponderance of evidence has warranted the independent publication of information, allowing the community to benefit from the information freely. There is still, in all probability, valuable information out there, held in secret, from which vital clues can be extracted with the correct expertise. We have hair that appears to be of primate origin, yet is not from any recognized primate species. While this is also reasonably convincing, the observations about recovering DNA from hair samples are a significant roadblock. A strong study of hair needs to be published for the community to review. Even if not peer-reviewed in a scientific journal, the value of existing expertise is lost in secrecy of groups seeking notoriety upon the ultimate discovery of the subjects. Ultimately, this is a destructive delusion. Whole genome mitochondrial sequencing would be the best approach to understanding new samples. Given changes in technology available since the SGP was done, this is easier now than it was back then. Of course, there would be few looking for trackways without the photographic evidence that exists. Some of this evidence is quite convincing, much is quite subjective. Whole papers could be dedicated to this subject, but I will summarize with this. Original photos and movies using chemical photography that is well exposed and well focused are highly valuable and quite rare. Digital photographs and digital video are quite limited in detail without proper optical work on site. Digital zooming does not amplify detail in the same way that magnification of emulsion can with chemical photography. There is precedent for species recognition by photograph without a holotype specimen. However, the species can be located in the wild and observed. Those photographs are clear and indisputable. Also, there is DNA from them that is indisputable. Hair, tracks, and more detailed photo and video evidence can be readily obtained. These things cannot be said of the subjects at this time. Until we understand the subjects well enough to be able to find them in the wild and record evidence of them that is genuinely clear and indisputable, along with the recovery of DNA evidence that is clear and indisputable, there is more work to be done to achieve these goals. Realize that the complexity of DNA means that actually recovering DNA will, in fact, be indisputable. DNA cannot be fabricated and has a level of complexity that will make actual evidence clear once it is recovered from the wild, and this truly has not yet happened. In fact, DNA alone would be acceptable to demonstrate that a species once existed, and may still actually exist when it is recovered. More on that later in this paper. When we understand the subjects well enough to find them in the wild, tracks and hair and likely DNA will be more or less readily recovered, along with photographic evidence, even digital. I am the Curious Cryptid, and this concludes the historic review of the subject of Sasquatch and DNA science. The next installment will address a number of misconceptions in the Sasquatch community regarding the science of DNA. These misconceptions are actual general misconceptions about DNA, but they specifically impact the Sasquatch community significantly.